take a look at that. Let's go ahead and take a look at what we need to know about our mutations. Okay. So when we talk about mutations, what is a mutation? Mutation is simply alteration in the genetic sequence or in the sequence of the DNA, right? That's what a mutation is. Any alteration that is, or any change that happens in the sequence of DNA is going to be known as a mutation. Now, mutations, I already told you guys, can be harmful, harmless, or they can be harmless or silent, whichever word you want to use, or they can actually be beneficial, right? When we talk about your harmful mutations, so harmful mutations, like I suggested, were the ones that actually cause diseases, right? They actually cause diseases. So those are your harmful mutations. Then we can have our silent mutations or harmless mutations. So those mutations that occur on the introns, right? I told you guys that introns are the non-coding regions, right? Introns are the non-coding regions. So those mutations that will occur on introns, they are basically silent. They're basically harmless. How are they harmless? Because they are occurring on the non-coding region. Like I explained to you guys in the last class, the non-coding region is removed in the process of translation. When we do the modification of RNA, we remove the non-coding regions. So it would be harmless. Or another possible scenario is that the mutation changes the sequence in such a way that the triplet code is still coding for the same amino acid. This is also, I told you guys, another possibility, right? Because one amino acid can have multiple triplet codes. One triplet code cannot have multiple amino acid. ACC will only be for one amino acid. But glycine can have multiple triplet codes, ACC, AGC, for example, right? And what can happen if, for example, if it's ACC and AGC, there's one mutation. It goes from ACC and it mutates to AGC. Now, both of these triplet codes are for glycine, right? So in that case, even though there has been a mutation, but the amino acid that will be incorporated will still be glycine. So that's also a silent mutation. A mutation has taken place, but there are no effects of it, or there are no actually in terms of phenotype, in terms of the phenotype of the patient, you will not see any effects of it whatsoever. And that's the reason for it, right? These are what? These are your silent mutations. Please keep this concept very clear that for one triplet code, there's only one amino acid, okay? But for one amino acid, there can be many triplet codes, not many, like 2 or 3, okay? So one amino acid can be coded by many triplet codes. So amino acid can be coded by multiple codes, but each code is only for that specific amino acid. So glycine can have ACC, AGC, but ACC is only for glycine. It won't code for methionine, right? That way. And so those would be your harmless. And then we can have beneficial as well. Beneficial mutations are mutations that are actually, for example, help example of this are the mutations that allow adaptations that better the chances of survival. All those mutations that increase the chances of survival are considered to be beneficial mutations, right? So if there's any adaptation that allows an animal to have a better chance of survival, to evolve, that's a beneficial, it's a useful mutation. Now, other than this, we can have chromosomal mutation. Chromosomal mutations are mutations that occur on a section of the chromosome. So generally, an entire section of the chromosome is going to be affected, right? Then we can have genetic point mutations. Point mutations are mutations that occur specifically on the DNA. So if, for example, I have my entire chromosome and a whole section of that chromosome is, that whole section of chromosome is being changed, or that whole section of chromosome is being, altered, that is a chromosomal mutation. But if the chromosome, the mutation is only happening on the one single nucleotide, that is known as a point mutation, right? Mutation of one single nucleotide would be known as a point mutation. So if I change one single nucleotide, one single nitrogenous base, that would be a point mutation. There's only one base that's changing. If the entire section of chromosome is altered, that's a chromosomal mutation. Now, in genetic mutations, we can have different types of mutations. We can have substitution. I taught you guys a good example of substitution mutation, right? What was it? Sickle cell anemia. When one nitrogenous base is substituted with another. And an example of this was your sickle cell anemia, right? In sickle cell anemia, what were we doing? There was an incorrect substitution. And because of that incorrect substitution, we were getting a different amino acid being incorporated into the polypeptide chain. And as a result, the structure of hemoglobin was changing. Other than this, we can have deletion. Deletion is when one of the bases is deleted from the sequence. This is a frame shift mutation. What do I mean by frame shift mutation? Now, when I'm talking about substitution, so in substitution, what am I doing? Right? I have, for example, my DNA sequence was A, C, G, C, A, C, G, T, A, C. A, T. All right. Now there has been a mutation. There was one mutation. In substitution, what's going to happen? The one of the nucleotide bases is going to be substituted or replaced with another. So for example, instead of getting A, C, G, instead of getting another C, there has been a substitution. Now I have what? Now I have A, for example. The rest of the sequence remains the same. A, C, G, T, A, C, A, T. So what has actually happened? What has actually happened is this, that the first triplet code is the same. The second triplet code has changed. The third triplet code is also the same. The fourth triplet code is the same. So only one amino acid will be different. Only one particular amino acid will be different, and that's this one. Instead of CSE, we now have AAC. So whatever amino acid was supposed to come that amino acid will not be correctly incorporated. The wrong amino acid will be incorporated into the polypeptide chain. This is a substitution mutation. Now, let's take a look at deletion. I will take the same sequence as above. A, C, G, C, A, C, G, T. Hold on, guys, give me a second. G, T, A, C, A, T. Now, there has been a deletion, which means one of the bases has been deleted. I will do the same one just for the sake of understanding. I will delete the same one. Now, there's a problem. What's the problem? In substitution, only one amino acid was being changed because only one triplet code was being changed. The problem with deletion is that the entire sequence gets changed. How? I have A, C, G. After that, the next C has been deleted. The rest of the sequence is A, C, G, T, A, C, A, T. This is one triplet code. This is another triplet code. This is triplet code number 3. And this is triplet code number 4. Now, the first one is the same ACG. But after that, instead of CAC, we have ACG. Okay, fine. Then what about the one after that? Instead of GTE, we have TAC. Then instead of CAT, we have AT whatever. This is a frame shift mutation. Why? Because of that one deletion, by removing this amino acid, I have shifted the triplet code completely. If there is a deletion, it affects all the triplet codes that come after it. Why? Because I have actually removed a nucleotide from the equation. Over here, I'm substituting. In substitution, only one particular base is going to be affected. That's it. In deletion, this is known as a frame shift mutation because the entire sequence has shifted. Why has it shifted? Because I've actually removed a whole number, a whole nucleotide from the middle, right? This is deletion. And this is the question that we were going over. Then the same thing can be seen for insertion. The same problem is seen for insertion. Right? Insertion is when one of the bases is inserted into the sequence. That's an insertion. And insertion has the exact same problem, right? That when we are going to go ahead and take a look, we are going to see what? Let me again take the same sequence and let's see what happens with an insertion. We have A, C, G, C, A, C, G, D, A, C, A, T. Let's say I decide that, okay, I'm going to add a extra nucleotide over here. What will happen to my DNA sequence? It will change and it will become A, C, G, C, C, A, C, G, T, A, C, A, T. Again, the first triplet code is the same. What is going to happen? But the next triplet code was supposed to be CAC. It's become CCA. The third triplet code was supposed to be GTA, but the third triplet code is now CGT, right? CAT was the last one, now it's ACA. So what has happened? Both of these are considered to be frame shift mutations. Why do we consider them to be frame shift mutations? Because by adding or removing one of the nitrogenous bases, I change the entire sequence of the triplet code, right? The triplet code literally shifts. So my triplet code will change. With substitution, there is only one triplet code that is changing. Why? Because the total number of your nitrogenous bases is the same. The total number is the same because I am simply substituting. I am simply replacing. But in deletion and insertion, I'm removing and adding. So that changes the total number of nitrogenous bases. And that changes the triplet code onwards. So after that mutation, all the triplet codes are going to be altered. So as you can see, this is known as a frame shift mutation. So these are the different type of mutations that you need to have an idea about. Making sense? We understand the basic concept of what's happening?
Yes. Yes, yes.
Good. Because this is exactly the answer we're going to give over here. The section of DNA molecule in figure 3.1 is part of a gene coding for a polypeptide. Base X shown in figure 3.1 is located in an exon. So because it's in an exon, you and I know it's not a silent mutation. It is going to be, it's most probably not a silent mutation on the strand of the DNA that is transcribed during protein synthesis. A mutation that results in the deletion of base X will affect the polypeptide produced. Explain how this deletion may affect the polypeptide during protein synthesis. This is where you guys are going to, this is exactly why I went over mutations with you guys. Because I wanted to, because you guys are going to see questions like these. So now let's go ahead and see how we can answer this question. Hold on, give me a second. 1 second. All right, so the points we're going to, let's go ahead now type out the answer, right? So how are you going to, what are we going to answer? We are going to go ahead and say, we're going to mix two topics to answer this question. One is the topic of mutations that we just discussed, and then we will also go over our topic of proteins. Please remember, whenever they give you questions about how a mutation is affecting a protein, you guys need to talk about both things, mutations as well as the structure of proteins. So you guys need to combine two topics into one answer. First, we will start talking about the mutation and then we will link it to the topic of proteins. So you can go ahead and say that deletion is a frame shift mutation, right? Which leads to a change in the triplet code on the DNA due to the removal of one of the nitrogenous bases, right? So first we're going to explain what it is, right? Then when you're going to go ahead and you're going to talk about it, then we will talk about why is this bad? Why is the change in the sequence of the triplet code bad? The reason it is bad is because now we will explain about triplet codes. Triplet codes are specific for one particular amino acid. The change in DNA sequence or the change in triplet sequence, whatever terminology you guys prefer, will lead to incorporation of the incorrect amino acid in the polypeptide chain. Now we will turn this on to relate this to the topic of our proteins. Okay. So then we will further continue and we will say the change, uh, the change, right? Uh, this, in fact, this results in alteration of the primary structure of our polypeptide or our protein. How? Because what was primary structure? Primary structure was the sequence of amino acids joined together by a peptide bond. The sequence of amino acid has now changed. So our primary structure has changed, right? This leads to what? This leads to change, or um this leads to different R groups being present, which affects the bonding that is seen within the polypeptide structure. Because what's going to happen, different R groups, different amino acids mean different R groups. Different R groups mean that your hydrogen bonding, ionic bonding, hydrophobic interactions, sulfur bridges, all of these bondings are going to be affected, right? As a result, the tertiary structure of the protein is altered, and then you can finally finish it off as hence a non-functional protein is produced. Or if you don't want to say non-functional, you can say that a protein with a different function is produced, whatever. So this is how deletion actually works. Got it? We understand the answer. This is all making sense?
Yes.
All right. Now, the next question says gene mutations occur in either introns or exons. Suggest the effect of a gene mutation in an intron. I just told you guys while discussing. What is the effect of gene mutation if it occurs on an intron, on the non-coding region of DNA?
It's a harmless or a silent mutation.
Yes, exactly. It's a harmless mutation. So way you're going to say is that it will have no effect on protein structure. Say it like that. It will have no effect on protein structure because introns are non-coding. Give an explanation. Because introns are non-coding. Okay, give me a second. Let me open up another question. Hold on. Okay, this is a really interesting question. The question says ACT can act on the stage of the life cycle of P. falciparum. So P. falciparum, they have talked about it before air, is that it causes malaria. It's a species of the plasmodium that causes malaria. It's known as P. falciparum. We're going to study about malaria, inshallah, when we do the topic of diseases. So ACT So ACT is the drug, one of the drugs that we can give to the patient. It's this is the full form of it. Artemisinin based combination therapy. So this is the therapy that we can give. Okay. So ACT is the therapy, and the pathogen is P. falciparum. ACT can act on the stage of the life cycle of P. falciparum that occurs within red blood cells. Inshallah, when we will do the topic of malaria, you will see that the pathogen of malaria, it has different life cycles. So some of the life cycle it spends in our liver, some in the red blood cells, some in the mosquito. So this therapy affects the pathogen when it is in the red blood cells. The cells of P cells falciparum in this stage are known as trophocytes. Okay, so the cells of the pathogen in the red blood cells, the name for them is trophocyte. Figure 3.1 is a photomicrograph of a blood smear. Some of the red blood cells contain trophocytes, and they've shown you, labeled it for you, that you see these small ring-like structures. These are all your pathogens. Now, they're saying PFK13 is a protein. So we are automatically going to be highlighting points as I read the question. So this is easy for you and me to solve. PFK13, what is it? It's a protein that has an important role in the development of trophozoite stage of P. falciparum. So we need this protein for the development of trophozoite stage, which means for the plasmodium to exist or the plasmodium to survive in the red blood cells, it needs this protein. Okay. The gene KELCH13 codes for PFK13. So KELCH13 codes for PFK13. PFK13 is the protein that helps the malaria survive in the red blood cells. Okay. Now, so the gene is KELCH13 and the protein is PFK13. And what's the function of the protein? Help in survival of the malaria. Two different mutations of KELCH13 known as F4461 and C580Y. So this gene, KELCH13, can have two mutations. Okay, one is F661 and one is C580Y. Were investigated to see if they were associated with partial artemisinin resistance. Artemisinin is the drug therapy, and resistance, like, you develop drug resistance, antibiotic resistance, bacteria develop antibiotic resistance. So they're basically seeing if mutation of your KELCH13 is going to help the pathogen develop resistance to the drug. That's what they're trying to see, because KELCH13 gives us the protein that they need for survival. So they're seeing, does the mutation in this gene, is that the reason that these malarial pathogens are able to develop resistance for the drug therapy? Is that the reason? Now, the question given to us after all of that extra knowledge, which we will see, using gene KELCH13 and mutation F4461 as examples, explain the difference between a gene and a gene mutation. Question is really easy. They simply want you to give the definition of what is a gene and what is a gene mutation. And then in examples, you can write down KELCH13 and F4461. Right? First of all, what is a gene? Does anybody know the definition of gene? Does anybody remember what's the definition of a gene? Does any remember the definition of a gene? When you're talking about a gene, So gene is, what is gene? Gene is a code for our protein. That's what a gene is, right? It's simply a code for our protein. A gene is simply a code on the DNA for the formation of a protein. That's a gene. And then you have to give an example using the Kelch13 and F4461. So between Kelch13 and F4461, who is the gene? Who is the gene? Kelch13. They have even given you the answer in the question. The gene is Kelch13. One, one definition, gene is a code on DNA that is for the formation of a protein or a sequence of DNA that codes for a protein. That's the definition. Example, Kelch13. And then you will give the definition of mutation. A change in the sequence of DNA is known as a mutation. Example, F4461. That's it. That's all we have to do. Give a definition of gene, give a definition of gene mutation, because we have to explain the difference. Right? We have to explain the difference. We give the definition of gene, we give the definition of gene mutation, and then we simply write down that Kelch13 is a gene and mutation, the example of mutation is F4461. Making sense? Understand how we're getting the answer?
Yes.
And then if you want, you can even give a little bit of add a little bit of the information over here. You can simply write down that when you're giving the definition, so you write down that gene codes for the production of a polypeptide or a protein. An example of that is Kelch13, right? Then you can go ahead and say a gene mutation is a change in the sequence of amino acid that leads to a change in the protein sequence, right? That's what it is. It's a change in the sequence of DNA that leads to a change in amino acid sequence that leads to a change in protein. And then you can add a few examples as well from this. When you're talking about mutation, you can say that in the mutation F4461, adenine is substituted for thymine. In the normal DNA, when we have normal Kelch13, it's thymine. The nucleotide we have after mutation is adenine, right? So the nucleotide after mutation has changed. So you can even use the information from the table that F4461 is the mutation in which thymine is replaced by adenine. And as a result, the amino acid is also changed, because in normal protein, it's phenylalanine, but after mutation, the amino acid is isoleucine. So this changes the structure of the protein. So you guys can even use examples from over here to make your answer better. Now, next they are saying in the investigation, the survival rate of trophozoites within red blood cells was determined. One second. In this investigation, the survival rate of trophozoites within the red blood cells was determined for two different concentrations of the artemisinin-based drug known as DHA. So the drug is DHA, and what they are actually doing over here is that they are trying to find out the survival rate of trophozoites. So what they've done is that they've taken a low concentration of the drug and a high concentration of the drug. And then they're checking to see how many trophozoites are going to survive, right? Then, as you can see, they have taken two different strains, A and B. So they've taken two different types of the pathogen, two different types of the malaria pathogen. They have done no mutation, then mutation with F4461 and mutation with C580Y. Both mutations as well as no mutation. They have considered all the scenarios. Now, this is the result that is in front of us. The first strain with no mutation, then the same strain but with F446 mutation, then again strain A but C580Y mutation. Then they took the other strain, strain B, and similarly no mutation, F mutation and C mutation, right? So they have compared all the mutation as well as two of the strands. And then they have treated, so what they've done is that they have treated all of them with a low concentration of the drug and a high concentration of the drug, and they have calculated the survival rate, meaning that how many of the trophozoites survived, right? Now, question says, state the main conclusions that can be drawn from the results shown in figure 3.3. So let's go ahead and take a look. What conclusions can we draw? First of all, we are going to start our answer with a blanket statement. Blanket statement means we will give a general observation. The general observation I think we can see is that for the low concentration, this is the low concentration. For the low concentration, the survival rate is a bit higher compared to the high concentration, right? At 700, the survival rate is very low. While at 20, there are still some trophozoites that are able to survive. This is a general statement. We have not gone into the intricacies. We have not gone into the explaining what we can actually see. We are just going over the general statement. So we will first start off with we're first going to go ahead and start off with our general statement. General statement, you can go ahead and say that the survival rate of trophozoites in the 700 nanomolar DMQ is lower compared to the 20 nanomolar per DMQ, right? That makes sense. That's our general statement, a blanket statement. Now we will slowly start to compare the results. We will say, let's take a look at no mutation. So the 700 nanomolar one, the 700 nanomolar one, there is no survival rate, right? No survival rate when for both strains with no mutation. So when both the strains have no mutation, when both the strains are showing no mutation, so at 700 nanomolars, we have a survival rate of 0. So we can go ahead and talk about that, right? Then we can go ahead and talk about our mutations. Then we can start talking about the mutations one by one. So if I talk about the mutations one by one, what am I going to say? Let's start with talking about our strain A F4461 mutation. The F4461 mutation, again, you will see that strain A has a higher survival rate compared to strain B. This is the value for strain A for both, for 20 and for 700. And this is my strain B. So if you will take a look, you will see that at both low concentration as well as high concentration, the FF461 mutation in strain A, same mutation, but in strain A, they have a greater survival rate compared to strain B, right? Then if we talk about our this mutation, CD580Y, so we will notice the same thing. Strain A with the same mutation seems to have a higher survival level at both concentrations, 20 as well as 700, low and high. At both concentrations, CD50 strain A has a higher value. Strain B has a lower survival rate. And this is true for all three scenarios. When there is no mutation, strain A has 3.15 survival at low concentration. Strain B only has 2.86. Even with the mutations, the survival of strain A is higher and the survival of strain B is lower. So we can add this point to our answer as well. At both concentrations, the survival rate of strain A is higher compared to strain B, regardless of the presence of mutations. Regardless of whether mutation is present or not, the survival rate of strain A is going to be higher than strain B, right? Then, when we're going to talk about it, let's compare the mutations with no mutation. So you can clearly see that the presence of mutation is beneficial to our uh, pathogen. Why? Because the survival rate is higher with the mutation as compared to without the mutation. If there is no mutation, for example, at 702 concentration, when there is no mutation, the survival rate is 0. But with mutations, there is still some survival rate that is present. So that's another point we will mention, that your strains A and B, both strains A and B have a higher survival rate when they have mutations. It doesn't matter what mutation, but as they have a mutation, they have a higher survival rate compared to no mutation, right? And finally, let's compare the two different type of mutation. We have F mutation and C mutation. As you will see, the F mutation has a lower chance of survival, 26 and 33. Even in strain B, if you take a look, 13 and 17. Come on to the high concentration, same thing. 0.73, 0.91, 0.53, 0.63. So our mutation F is, mutation C is more powerful. Or how will we say it? We will say that both strains with mutation C580Y have a higher rate of survival compared to strains with mutation F4461, right? So these are our final conclusions. This is our final answer. Making sense? We understand how we are going to give answers like these, because you do get a lot of questions about tables and you guys should know how to write down the answers related to your tabulated data. Making sense? If anybody have any confusion, would anybody like me to go over anything? This is all making sense?
Yes.
All right. Now give me a second. Hold on. All right, take a look at this. The cell cycle concludes mitosis. Okay? What are the features of this type of nuclear division? So, what are the features of mitosis? Form cells of equal size to the parent cells, forms genetically identical nuclei, semi-conservation replication of DNA. Which is correct and which is incorrect? Or are they all correct? What do you guys think? Is statement number 1 correct? We will get a cell of equal size to the parent cell? Yes or no?
Yes.
All right. What about the second one? Forms a genetically identical nuclei. Is that true?
Yes.
What about semi-conservative replication of DNA? Is that true?
Yes.
All right. Now, when you're going to talk about it, they're asking you about mitosis. Mitosis. You only have to give answer to mitosis. Does the replication of DNA occur in mitosis or does it occur in the S phase of cell cycle? It occurs in the S phase of cell cycle. DNA replication doesn't happen in mitosis. You will replicate the DNA and you will make sure it's ready for division before mitosis, right? The S phase of replication is when we actually, S phase of cell cycle is when we actually make a copy of DNA. The statement is correct. We do get semi-conservative replication of DNA, but not during mitosis. That is during the S phase. Similarly, form cells of equal size to the parent cell. Not during mitosis, this is cytokinesis. When the cell will divide into two, that is mitokinesis. Mitosis is simply the nuclear division. I'm simply dividing the nucleus into two. The cell still has to divide into two and it will divide into two in when cytokinesis. So first point is correct, but it doesn't happen during mitosis. It happens in cytokinesis. Point number 3 is also correct, but it doesn't happen in mitosis. It happens in the S stage of cell cycle. The only thing that actually happens during mitosis is you get a genetically identical nucleus. Hence, for mitosis, only option 2 is correct. This is why we will always pay attention to the question. We will always highlight. We will always see exactly what they're asking us and make sure our answers are according to that. A student observed the cells in the growing region of an onion root and obtained the data shown. What percentage of cells contain chromosomes that appear as two chromatids? Let's first of all mark. Where do you think we will see two chromatids? In interphase, are we going to see chromosomes with two chromatids? What do you guys think? What do you guys think? Okay, we have to calculate the percentage. For percentage, we need the total number of cells. Okay. To find out the total number of cells, let's first do one thing. Let's calculate the total number of cells. Let's add 14 plus 14 plus 11. We have 1,000 cells. The total number of cells are 1,000. Now, tell me, in which stage do you think I will see two chromatids? Interphase. Are we even going to have chromosomes in interphase?
No.
No, there are no chromosomes in interphase, right? You only have chromatin. What about prophase? Will we have chromosomes in prophase?
Yes.
Yes, correct. What about in metaphase? Not only chromosomes, but chromosomes that will appear as two chromatids, that will appear like this. Okay? Metaphase. Will we have chromosomes that will appear as chromatids, two of them? Yes or no?
Yes.
Yes. Why? Because in metaphase, all of them are going to arrange at the equator. What about anaphase? Will I see chromosomes as two chromatids?
Yes.
No. Why? Because now the chromosomes have split apart from each other and they're now traveling to the opposite pole. They're no longer attached to each other, the chromatids. They're now moving to the opposite side. So even though I can see the chromatids, but I won't see them together as a chromosome. In prophase, I will see a whole chromosome with two identical chromatids. In metaphase, I will see the chromosomes arranged on the equator with two identical chromatids, but not in anaphase because I have now pulled them away from each other and they're no longer together. What about telophase? Will I see them or won't I see them?
You won't?
You won't. Same logic because now the chromatids have gone to the opposite poles of the cell and the nuclear membrane has formed around it. Right? So we will see this in prophase and metaphase. So we will, in prophase we have 73 and in metaphase we have 16. 73 plus 16 gives us 89. So 89 from 1000 cells, only 89 cells will have chromosomes that will appear as two chromatids. So to take out the percentage, we will do 89 divided by 1000 multiplied by 100. Our answer comes out as 8.9%. That's option B. Got it? Making sense? We understand how we're getting the answer?
Yes.
All right. Okay, guys, we're going to go ahead and stop it over here for today. Now, inshallah, in our next class, we're going to go ahead and start off with